and circulatory systems are, then go over 3 animals in specific, which are the shark, the rat, and the squid. Next, we're going to describe the dissection of a sheep's heart and then cover a disease that can affect the respiratory and circulatory systems. To conclude, we're going to describe the evolutionary history of the respiratory and circulatory system in our organisms, as well as compare their evolutionary histories using a cladogram. So, what are the respiratory and circulatory systems, and what do they do? To begin, the respiratory system is a network of tissues and organs that work together to enable the process of breathing, which is when you inhale oxygen and exhale carbon dioxide. And organs that are commonly found in the system are your nose, your mouth, your lungs, your trachea, and your gills. Next, the circulatory system is a network that composed of heart and blood vessels, and these work together to deliver oxygen, nutrients, and hormones to structures like your muscles, your organs, and tissues all throughout your body. And structures that are found in this system include the veins, the arteries, the aorta, arterioles, and venules. Now we're going to get into our organisms.
Hi everybody. So the first animal our group will be investigating to study the respiratory system is the shark. The type of shark that our group studied is commonly referred to as the spiny dogfish or Carolina dogfish. It is referred to as the spiny dogfish because the dogfish contains spines which flare out when a predator is nearby. The scientific name for our shark is the Squalus acanthias. Sharks are a type of cartilaginous fish belonging to the class Chronodictyes. They are known as cartilaginous fish because instead of containing bone in their body, they contain cartilage. Cartilage is much more flexible and durable than bone. Bone is more tightly packed together. The reason why this is important is because since cartilage weighs less and is more flexible, it helps sharks to stay buoyant. Additionally, sharks are aquatic vertebrates. The type of biome sharks live in is the temperate ocean. To the right, I have attached an image of a worldwide map showing the region of temperate oceans in the green area. It's like the green closer to the gray and blue areas. The geographic range in the Earth where temperate oceans lie and where Carolina dogfish are found include the North Atlantic Ocean, the Pacific Ocean, and the Gulf of Mexico. Here is another world distribution map for the spiny dogfish. This map is more specific than the previous map because it labels the presence of spiny dogfish. This occurs as scientists have tagged dogfish across the world, and using these tags, they collect data to measure their presence using technology. The red measures the presence of the spiny dogfish. The ecological niche of a spiny dogfish is really unique as they are bentopelagic. This means that the spiny dogfish lives near the bottom and also moves upward in the seafloor. They occupy shallow waters of continental shelf bays and estuaries. Additionally, Carolina dogfish are known to be demersal, which means that they are bottom dwellers. They also have migration patterns. They move north in the spring and south in the fall to adjust to the change in weather. Here is another geographic map of the change in migration patterns. The red identifies spiny dogfish tags when they migrate to the north. Notice that the red tags are more clearly aligned upwards compared to the black tags. The black tags represent the migration patterns to the south. Dogfish are called dogfish because similar to how dogfish hunt in packs, these spiny dogfish also swim in large groups known as schools. The reason that they do this is for protection while hunting. By swimming in large groups, they are less likely to be predated on by nearby animals. Additionally, this assists with sexual selection as males tend to pair up with the females that display the biggest and baddest behavior or aggressive behavior. Here is an image of the Carolina dogfish sharks swimming in schools. Notice how they are towards the bottom of the seafloor, like you could see all the sand. This indicates their demersal property where they stay towards the seafloor. Additionally, they have multiple roles in their ecosystem and environment. First of all, they are keystone species. This means that the Carolina dogfish regulates population sizes of smaller predators such as cod. This occurs so that way these codfishes do not overgrow and there is an abundance of one predator. Additionally, they are generalists in food choice. Generalists are organisms that have a wide variety of food and basically eat whatever they can find. Typically, the spiny dogfish consume small predators such as shrimp, crab, jellyfish, and squid. Additionally, they are scavengers, which means that they eat dead remains of organisms. Typically, the Carolina dogfish eats dead remains of shrimp and crab. By doing this, ammonia is recycled back into the environment through the decomposition of these animals. This is important because ammonia is used in the nitrogen cycle as nitrogen is cycled back into the environment. Nitrogen is important as it's a main building block of proteins and nutrients for plants and other organisms. Imaged here is a food web displaying the Carolina dogfish's role in the food web in their population and community. The next topic I would like to discuss is reproduction. Carolina dogfish typically reproduces through internal fertilization. Their eggs are ovoviviparosis, which means that the eggs are in the mother and they give live birth. The gestation period is really interesting because it's 2 years, which is one of the longest periods in the animal kingdom. They produce 5 to 12 pup sharks per birth. Now, this is important because typically a lot of cartilaginous fish produce up to 50 or even more than 100 eggs. But it's important to note that the Carolina dogfish produce 5 to 12 pup sharks per birth because they are a case-selected species. This means that since they produce a fewer amount of offspring, they can devote more parental care to each offspring. Whereas a fish that gives birth to, let's say, 200 eggs won't have enough time or resources to give every egg the amount of care it needs. Additionally, they display logistic growth patterns, which is imaged to the right. Now, human impact has played a big role in the development of the spiny dogfish. Their population first peaked in 1974 and declined in the 1980s. Now, this decline is due to the human use of Carolina dogfish for food in the European industry. For example, the spiny dogfish are used to make fish and chips, and they're also used as for oil. Now, commercial fishermen have been targeting mature females because they grow larger than males. And in the 1990s, because of all of the commercial fishing that was occurring for these females to be a food source, there was over a 75% reduction in female sharks. This statistic is very dangerous as it could potentially pivot the spiny dogfish to being endangered. Luckily, human action has been taken since to reduce the number of Carolina dogfish species decrease annually. In 2001, the ASMFC voted for an emergency action to close fishing of the vulnerable dogfish. At this time, because of the 75% reduction in female sharks, the spiny dogfish became classified as a vulnerable species. 2 years later, in 2003, there was a fishery management plan implemented which lowered the quota of dogfish to be hunted. This data was collected, the data was basically used through scientific data to determine how much dogfish could be fished so that way these species could grow and not be considered a vulnerable species. Additionally, they were listed as vulnerable due to overfishing. So some important adaptations I'd like to go over include the bow shape. The spiny dogfish can curl into a bow, as shown in this image. If you could see, it's almost like a sine graph, like the tail. And this bow shape is important because they could strike out their spines to injure predators. And the reason why they're called the spiny dogfish, as I prefaced in the beginning, is because they have sharp venomous spines in front of each dorsal fin. The dorsal fin is the top two fins closer to the head of the Carolina dogfish. This protects the spiny dogfish from sharks, seals, and cod, which are usually their main source of predators. Their fusiform body shape, which means that it's narrow towards the ends, reduces drag. Reducing drag is important because it leads to faster speeds in handling. Additionally, because their skeleton is made out of cartilage, which is a common trait for cartilaginous fish, it is more lighter and flexible than bone. Being lighter helps these sharks stay afloat and also reduces the amount of energy they need to expend. Their lightness also allows them to make sharp turns. Next, their skin is covered with millions of dermal denticles. These are tiny teeth pointing backward that help their movement in their biome by reducing drag. This allows sharks to swim faster to run away from predators. Imaged here is an example of dental dermicles shown. As the shark grows, they shed their denticles and replace them with larger denticles annually. Now, a very important adaptation that these spiny dogfish have evolved include teeth. Teeth are made out of enamel. They have 3 types of main teeth: flat crushing teeth used to eat shellfish, pointed teeth for gripping fish, and sharp serrated teeth for larger specimens. Here is an image. In our shark that we dissected, we noticed that our shark presented the feature of having grinding teeth. This means that we could potentially predict that their source of food would be prey with shells such as the bow-mouth and guitar shark. Another adaptation is gills. Gills are important because these cartilaginous fish living in the ocean possess them. Unlike humans, sharks do not breathe through their nose. They have their gills to take in water, which pass over the gills, and here oxygen is absorbed into tiny blood vessels and transported around the body. This process is known as ram ventilation. In summary, to keep sharks buoyant, which is a crucial feature given that they're sea life, they rely on their light skeleton composed of cartilage, their lift effect, and their large livers that store low-density oils. Since they're low density, it contributes to their lightness. These large livers make up 25% of their body weight. Another important feature of the spiny dogfish is their ability to detect electric currents. This feature is known as electroreception. Image to the left, as you can see, the tiny muscular movements in the small fish current through the ocean and used by the sharks to detect prey and predators. This occurs because at the site known as the ampullae dei Lorenzini, which are tiny black holes found in the upper jaw and snout, is filled with electrically conductive jelly. And at the bottom of these holes, once the electric current passes through, are lined with cilia. Now these cilia serve as receptor proteins, which basically conduct the electric signal and transmit it to the nerve cells, so the sharks can feel the electric current. The flow of electric current stems from the jelly of the Lorenzi de ampullae to the cilia and then to sensory cells, which automatically transfer the signal to the brain. This allows sharks to detect whether there are predators nearby so they can easily escape. To now continue with our investigation of the respiratory system of a spiny dogfish, I would like to introduce the respiratory organ system. The pink here represents the system that makes up the heart. The parts that make up the heart of a spiny dogfish include the conus arteriosus, the atrium, the ventricle, and the sinus venosus. Note, the heart of a spiny dogfish is 2-chambered, unlike a human, which has 4 chambers. The light green represents the gills, the primary point of gas exchange through which water enters the shark. Next, the purple represents the arteries in which oxygen travels through. The first part I would like to talk about is the gill arches. These gill arches, which are basically rows housing gills, are basically where gill slits occur. And these rows contain little thread-like filaments called gill filaments. These are thin, thread-like structures responsible for gas exchange. We also have our internal gill slits, which are basically an opening in the pharynx where the gills are housed. Now, this is really important because gills serve as the site of gas exchange. Oxygen is exchanged here for carbon dioxide and blood is oxygenated. It's important that blood is oxygenated so that way it can travel to the rest of the parts of the body and power the shark to live. Next, near the esophagus of the shark's system, we have the gill rakers. These prevent food from entering the gills. They have specialized shapes and structures, which basically, when you fold the shark, it would prevent the food from ever entering the esophagus. Next, I would like to dive into the heart. First, we have the conus arteriosus, which is labeled. This is basically the section that carries blood to the ventricle. The sinus venosus carries blood to the atrium. And the atrium here carries blood to the gills. The ventricle is responsible for heart pumping. This heart pumping movement is it's basically not controlled by anything. It's all simultaneous. We also have our efferent branchial arteries, which are responsible for transporting blood to the body and create sound.
Human impact. The spiny dogfish population peaked in 1974, followed by a sharp decline in the 1980s. This species of shark is widely known for its use in the British dish fish and chips and for its oil. It can cause great damage when these are caught in commercial nets. Commercial fishermen mainly target mature females of this species as they grow to larger sizes than males. In the U.S., the dogfish fishery heavily increased in the 1990s, which led to a 75% reduction of mature females and subsequently a record low number of pups born. In 2001, the Atlantic States Marine Fisheries Commission voted to extend an emergency action to close fishing of the vulnerable dogfish in state waters in response to this 1990s decline. In 2003, the ASMFC implemented a plan to lower the spiny dogfish quota to a level supported by scientific data. The spiny dogfish is currently listed as vulnerable by the World Conservation Union due to this overfishing.
Our next specimen is the rat. To classify our rat, it is a Wistar rat or an albino Norway rat. It gets its name Wistar because of the company named Wistar that set the precedent of using the albino Norway rat as a lab rat in experiments. Its scientific name is Rattus norvegicus and it is a mammal. The biome that these Wistar rats are most commonly found isn't actually even a biome, it's laboratories. And this is due to several rounds of artificial breeding done by the Wistar company to create populations of Wistar rats that are low in genetic diversity. And low genetic diversity is an advantage for researchers because it ensures that in experimental results, genetic diversity won't be affecting it. Non-albino Norway rats, however, can be found everywhere, in every continent except for Antarctica, because Norway rats are commensals with humans, and Antarctica doesn't have humans. Now, they can also be found in savannas, grasslands, and chaparrals, which are terrestrial woodlands with dense woody vegetation. And they can also be found in scrub forests, which are dense, low-lying woody vegetation forests. Norway rats have a highly developed sense of smell, which is crucial for finding food. They are also great climbers, jumpers, and swimmers, as they can jump as high as 3 feet into the air and they can swim on days on end to find new shelter. They can also withstand extreme temperatures with their fur, as they can easily release heat as well as keep heat. However, as explained before, they can't survive in Antarctica despite being able to withstand extreme temperature because of the more important fact that they're commensals with humans. Another physical adaptation that Norway rats have are that they have excellent whisker dexterity, which means they can move each whisker individually like a human's finger. If you look at the top right photo, you'll see the orange and yellow diagram of the rat's nose. And those are essentially receptors that allow the rat to move each whisker individually. And these whiskers, along with the paws, actually allow the Norway rat to sense minute vibrations, which allow the rats to communicate with each other. These whiskers also help the rat be aware of its spatial surrounding due to their poor eyesight, which makes up by helping these rats with their sense of feeling and sensing. They also have physical adaptations with their paws and tails because their paws and tails allow them not only to balance but also regulate their body temperature. They also have sharp teeth called incisors, which are non-stopping growing teeth. So the more that their teeth are used, they get grinded down, but the teeth will continuously grow. And this is essential for the rats because they have a wide range of diet. So they'll eat anything hard from nuts to soft food. And so with such heavy diets, their teeth will get grinded down and they need their teeth to be replenished. A behavioral adaptation that they have is that they have high intelligence, which is actually the reason why Norway rats can be found anywhere across the world because they're able to adapt to any environment that they need. They can change their behavior, their diet, and where they seek to find shelter. They also have cautious tendencies because of their poor eyesight, which means that they use their whiskers, paws, and tails to essentially create a memory map in their brain to memorize what their environments feel like and like vibrate as. And if any one of those feel out of place, this causes their cautious tendencies to kick in and they'll only remain in the places that they know are safe. Looking at the habitat of the Norway rat, you'll find them in both temperate and damp environments. As for temperate environments, you'll find them in woods, forests, meadows, fields, and tropical islands, such as the bottom left image below where you can see a little rat hiding away in shrubs. As for damp environments, you'll find them near riverbanks, coastlines, and wetlands, such as the coastline shown in the top left photo, as well as human environments such as sewage, garbage dumps, and basements. And you can see in the top right photo, there's a Norway rat residing in a sewage pipeline. As for their tunnels, they're created through burrowing, and at the center of their burrow, the Norway rats will dump any soft vegetation that they'll find, or materials such as sticks or food that they'll find from scavenging and foraging. And their entrances and exits are called bolt holes because they're convenient for rats to use when they escape from predators. Now, their diet has a wide range. It consists of berries, seeds, nuts, and etc., as well as anything digestible like grains, shrimp, snails, mussels, insects, and bird eggs. You'll see photos that show all such of these foods. And their diet is also strongly influenced by their habitat, which is explained by their behavior explained earlier of how they change their own behavior and their diet and their shelter seeking habits just to survive in the environment they're put in. As for their reproduction, they can start reproducing as young as 4 weeks old, and when they get pregnant, they're typically pregnant for 21 to 23 days. And as soon as they give birth, they can get pregnant immediately 24 hours later. And when they give birth to large litters, almost 16 to 13 offspring at once. And this results in the large numbers of rats being reproduced from one mother rat that you'll see, typically around 400 rats. And they lean towards R selection species. They lean towards being an R selection species because they're smaller animals that produce large numbers of offspring at once, but they don't exactly give much care to their offspring. So relations with animals, what does this look like for the Norway rat? So Norway rats are prey of felines, birds of prey, and weasels. So if you look at the top left image, you'll see a bobcat, which is a feline that the Norway rat is a prey of. And if you look at the photo to below it, you'll see an owl, and to the next is a weasel, which are also predators of the Norway rat. However, Norway rats are also predators of other species, such as crabs, small turtles, and rodents. Now, you can see competition between rats and mice as they're such similar organisms that inhabit similar environments and compete for similar resources. So the mice is the organism that rats compete with the most. Rats are also commensals with humans, as human development allows for there to be many damp environments and a lot of food droppings for the rats to feast on. So you'll find rats often infesting human environments, such as your basements after there's been a downpour of rain, because there's a damp environment created. And you'll also see that there's mutualism between kangaroo rats and harvester rats. So kangaroo rats, with their burrowing, provide natural shelter for harvester rats, ants, but harvester ants also provide food that they harvest to for the kangaroo rats in their central collection of the burrow materials. You can also see parasitism between rats and Syphasia murus, which is a type of pinworm that deposits its own eggs inside of the rat, and then these eggs they hatch and it just creates a colony of these parasites inside of the rat and feast on the nutrients of the rat, causing the rat to slowly die. Specific roles that the rat has are soil aeration and seed dispersion. So, what I mean by soil aeration is that when these rats are burrowing and are tunneling when burrowing, they'll mix different levels of soil, and this causes the spread of a widespread reach of different elements such as nitrogen, phosphorus, sulfur, carbon, and oxygen to spread around the soil, and this causes soil to be fertile for better plant crop planting, and it also allows for better water permeation, meaning better irrigation for crop harvesting. They also help with seed dispersion, so gene flow of seeds, because they can store many seeds at once when they're foraging, but as they're traveling they often drop many of these seeds, which results in the gene flow. So, as a summary of everything discussed, the important roles that these Norway rats have are that they're opportunistic, omnivorous scavengers. As you can see, they prey also on other organisms, not just vegetation. They'll eat their own kind, as I've stated. They eat other rodents. They'll even eat their own kind when they're desperate enough. They're also foragers, as seen by their herbivorous diet. Next, we have our rat dissection to view the respiratory and circulatory systems. To begin, we have the external nares depicted on the right, which are basically your nostrils, and these nostrils allow for oxygen to be inhaled and exhaled. And the nostrils on the rat is smaller than the ones we have as humans because they have overall smaller body proportions, so they need to block out large aerosols such as smoke and dust, and they also have not really complexity, more of an adaptation, as stated before, their whiskers, which again, they are highly developed. And then we have the nasal passage and the nasal cavities. This nasal passage and nasal cavities is where you'll find air being filtered as they enter the airway. And so you have a septum which separates the left cavity from the right cavity. And inside each cavity, you'll find there's three regions: the nasal vestibule, the nasal conchae, and the olfactory organ. In the nasal vestibule, this region has hair, sebaceous glands, and sweat glands, and these glands produce substances which lubricate the lining of the cavities, and these substances help catch the bacteria as the air passes through. And then the nasal conchae are scroll-like projections off the side of the nasal cavities inside your nose, and these help increase the surface area for air to pass and be detoxified through. Then there's your olfactory organ, which is simply an organ that helps receive the sensory information of smell. There's also your vomeronasal complex, which are just organ, blood vessels, cartilage, and glands. And this complex gives rats the ability to smell and track scents such as pheromones when mating. And they also contain vomeronasal sensory neurons, which express receptors that send an information to the olfactory bulb in the brain. They also have complex turbinates, which are tiny structures in the nose that help cleanse, humidify, and heat air as they pass through the nasal cavity. And they also allow for enhanced depositing of aerosols, meaning they catch air. In this more complex turbinates in the rats allow for aerosols to be caught at a faster rate and more efficiently, basically, because again rats are smaller organisms, so they need to be able to defend themselves from all the large particles that can be found in our environment. Next, we have the olfactory epithelium, which is the smell tissue, which is found more common in rats as it lines 50% of their nasal cavity. And olfactory in this name just means like being the sense to smell. This epithelium tissue is actually pseudostratified neuroepithelium, meaning brain tissue. And there's three cell types, one being basal cells. They're globular and horizontal in shape, and these cells give the rats the lifelong replacement of the olfactory epithelium. And then there's the intermediate cell type, which contains both immature and mature olfactory neuron receptors, and they help replace these receptors as time goes by. And then there's the sustentacular epithelium cells, which are columnar cells, which help support the base cell and intermediate cells in their function. And these sustentacular cells are actually also produced by the base cell cells, but then they also help them replenish. And basically, this olfactory epithelium works in rats by trapping odor that passes across the cilia and sending information to the brain. Next is the pharynx, which is essentially just the throat in the rat, and this is where food and air pass through. And in this image, we annotated a more superficial area. This isn't where the pharynx actually is, it's underneath. However, the group couldn't get the jaw to open because of how strong the muscles are in the rat. But we have provided a diagram later on to explain where it is more essentially. There's also the glottis, which is the vocal cords that reside in the vocal box, and these create the sounds that the rat uses for communication. But there's also another structure which with a similar name but a different function called the epiglottis, and this is the flap between the pharynx and the trachea that prevents food from air from entering the wrong tunnels. And so for the respiratory system, this prevents food from going down the trachea. Otherwise, you could choke. So here are the diagrams mentioned earlier. You'll see on the left image that you'll see just how close in proximity the pharynx, the glottis, and the trachea is. Somewhere in between the trachea and the pharynx and but right before the glottis is where the epiglottis is, and that's where the flap essentially opens and closes for food or air to pass through. And if you see on the right image, you'll see just how small the epiglottis is. Next is the larynx, which is the voice box itself, and this, along with the glottis, is where the sound for communication is created. And in rats, there's a special complexity called the ventral pouch. This has two functions. One, it helps with detoxifying the air that filters through, as it provides additional collection of toxic substances before the air reaches the lungs, but it also aids in communication because it resonates and amplifies any of the vibrations created by the vocal box. So next we have the trachea, which is just the passageway for air to travel to the lungs. And there's some complexities here, such as pseudostratified ciliated columnar epithelium, which are tissues that line the nasal cavity and the inside surface of the trachea and bronchi. And what these ciliated cells do is that, along with goblet cells, which are structures found inside of the ciliated cells, they secrete mucus. The mucus and cilia on the surface work together to trap any bacteria to provide additional filtering of air as the air is traveling towards the lungs. And this also helps with the mucociliary escalator, which is when mucus and cilia help travel mucus all the way up towards away from the lungs, and this is to prevent any bacteria from reaching the lungs. And this causes a reflex where, as humans, we will have the instinct to hack our mucus and we cough it up and then we swallow it, and when we swallow, the epiglottis will actually open and close the trachea and then open the pathway for the mucus, the coughed up mucus, to go back down and be digested to stomach acid, so the bacteria is all broken down. Next, we have the bronchi, which is just an extension of the trachea. However, unlike the trachea, which only collects air to be traveled down to the lungs, the bronchi is actually what distributes it because it directly connects to the lungs, unlike the trachea. However, you can't visualize this in the dissection image because the group was fearful that if we tried moving the heart to view the bronchi, the heart itself would rip. But we have a diagram later on to depict how the bronchi looks. And some complexities here are that there's serous cells, which are cells that line both the trachea and the bronchi, and these help with host defense because these cells secrete mucus that contain water, antimicrobial compounds, and enzymes that help latch on and break down bacteria found in the air. Now, this bronchi also has monopodial branching. This type of branching is when the bronchi branch down with two parent stems like this, and then there's child bronchi that branch off of each stem. And this is different compared to humans because humans have dipodial branching where each branch will branch off of one another. There's no parent branch. This influences inhomogeneity, which is essentially the lack of uniform distribution of air towards the lungs. And this can be explained by the different count of lobes in each side of the lung in the rat. If we look at the bronchi diagrams, you can see behind the left pulmonary artery and the right pulmonary artery is where the bronchi are situated, and you can see that they connect directly to the left and right lungs. And this is actually a picture of the rat lung itself, which we will discuss a little bit more later. And on the right image is the monopodial and dipodial branching differences. On the left of the right image is monopodial, where you can see that there's two parent branches which have children branches branching off of them. And on the right, you can see how each branch branches off of one another. So next we have the lungs, and so first, as stated, the air travels through the trachea, then the bronchi, and then the lungs. And in the lungs, this air is dispersed amongst alveoli, which are air sacs where oxygen is exchanged into blood, and carbon dioxide is exchanged out of blood and ready to be exhaled. And there's also an air-blood barrier in the lungs, which is a barrier made out of epithelial cells to separate blood from air to prevent them from mixing. And in rats, this barrier is thinner because it makes the gas exchange more efficient. And in the rat lungs, as stated before on the last slide, left pictured a diagram, the difference of number in lung lobes is different in the rats. As you can see, the right lobes, there's 4 of them, whereas there's only 1 lobe in the left lung. And this is unique for the rat because you don't really see that in any other mammal. And this makes sense because of the rat's size. So because the rat is smaller, its heart is going to take up a little bit more space, and the heart is situated towards the left side. So as the heart is beating, there needs to be less stress on the left lung when the heart is pressing against it. So to combat that, there's only 1 left lung to perform gas exchange work. But to compensate for this, on the right lung, there's 4 instead of how you may see typically 3 in other mammals. And this helps increase the surface area and volume of the lungs for gas exchange. You'll also see BALT tissue, which is bronchus-associated lymphoid tissue, and these tissues are related to the immune system of the rat. And these tissue can be found next to major airways in rats, and this tissue also takes up antigens from airways, and they also initiate local immune response. And this is important because this helps the rat keep its immune system functional against infectious diseases, tumors, and allergens. And they also help maintain memory cells in the lungs, so the lungs will remember how to defeat those infectious diseases. Next, we have the diaphragm, which is a muscle that separates the thoracic cavity from the abdominal cavity. And this thoracic cavity is basically the cavity where in your upper body you have your lung and your heart and your rib cage, and this muscle separates it to protect the functions of these organs from the rest of your intestines and your liver and your kidney. And this diaphragm muscle helps with inspiration and expiration. And what this means, inspiration is when you inhale, letting your air fill your lungs, and exhale, letting your air leave the lungs. And it does this because as the diaphragm contracts, if you took in a breath yourself, you'll see it contracts and it makes space for the lungs to expand. But as you're exhaling, this muscle releases, it relaxes, which lets your lungs to sink back into a relaxed position. So next we have the heart's rat, which is the blood pumping organ, and the heart is made out of cardiac muscle, which means and cardiac muscle is also involuntary, which means you don't have to think about how the heart is going to beat, it just creates your natural heartbeat. And in the rat, the myocardium is unique because it doesn't have a plateau between the action potential and the contraction phase. And it also has a different length of action potential itself compared to humans. Next is the protective membrane called the pericardium, and this membrane protects the heart from friction against the other organs that the heart is surrounded by. And the rat's heart is four-chambered, meaning it has two atrium, right and left, and two ventricles, also right and left. And so the atrium is this cavity that collects blood from veins from all around the body. And then the blood from the atrium is pumped down into the ventricle, and then the ventricle forcibly pumps back this blood to the rest of the body. And you can find catalase, the hydrogen peroxide breaking down protein, in rat heart mitochondria as well as the rat's lungs. And it has an antioxidant effect because it helps prevent damage from H2O2 accumulation. And without the function of catalase in rats, it can lead to ischemic heart disease, which is when there's reduced blood flow to the heart's muscle in the rat. So how does the circulatory system of the rat relate to its lifestyle? To begin with, it has a higher heart rate of around 260 to 450 beats per minute, which is significantly higher compared to humans. And this is due to their high levels of activity at night, being foragers and scavengers, trying to get as much food as they could get. And this also contributes to them being endotherms, and this means they're warm-blooded and that they regulate their own body temperature. And because they're so active at night, they release a lot more energy, so the heart pumps a lot more to produce more energy to sustain themselves. And scientists also suggest that heart rate, it correlates to how long a rat lives. So scientists suggest that the lower the heart rate of a rat, they have a longer lifespan, and vice versa. If they have a higher heart rate, they live lower lifespans. And and rats in general circulate less blood volume because they're smaller animals, so they don't have as extensive of a need for oxygen as humans maybe, so they circulate less blood. And now the respiratory system and how it affects the rat's lifestyle is that breathing and metabolism is heavily influenced by how rats sleep at day and night. So as stated before, rats are active during night and they sleep during day. However, if they can't sleep properly during the day, it can affect how well they can breathe at night. And so this in turn causes a cycle of implications, and if it affects their breathing during the night when they're scavenging and foraging, then they won't have the necessary resources to sustain themselves with nutrients during the day, which might cause more sleeping during the day to sustain energy. So how do the respiratory and circulatory systems actually work together? Well, first we have to separate the veins that carry deoxygenated and oxygenated blood. So first we have the right, left, and inferior vena cavas. The right superior and left superior vena cavas bring all the deoxygenated blood from your head and neck down to your heart, whereas the inferior vena cava brings all the blood from your feet up to your chest back to the heart to be reoxygenated. Second, we have the blood veins that carry all the reoxygenated blood back all the way to for the rest of the body to use for important processes such as the ETC. And this is actually the reason why rats are studied so much. It's because their circulatory and respiratory systems are so similar to humans. And so if we look at the aorta, it's the largest blood artery in the entire body for both rats and humans. And the aorta splits into the innominate artery and the aortic arch. And what these do is that they carry reoxygenated blood down to the brain, neck, shoulder, arms, and forelegs of both the right and the left side of the bodies, both doing their separate jobs. And if and whatever blood that the innominate artery and aortic arch cannot supply to the rest of the body, the dorsal aorta will do it. And this dorsal aorta is basically an extension of the aorta down your back all the way down towards your legs, and it carries all the reoxygenated blood necessary down there. So now putting all these veins together with the understanding that we first have pulmonary circulation, which is the circulation of blood between the heart and the lungs when deoxygenated blood is carried back to be is transported to the lungs to be reoxygenated. So if we look at the main steps, we'll see that the three vena cavas enter the right atrium to return deoxygenated blood, and then from this right atrium, blood is pumped into the right ventricle through the tricuspid valve, which is a valve that separates the atrium from the ventricle and prevents backflow from the ventricle to the atrium. And then this blood is transported to the pulmonary trunk, which eventually branches off into pulmonary arteries. And these arteries carry the deoxygenated blood back to the lungs for reoxygenation from the alveoli, the air sacs that extract the oxygen from the air that's being breathed in. And then there's the second circulation called systemic circulation, and it's called systemic because it's the circulation of all the reoxygenated blood that's transported from the lungs to the heart back to the rest of the body. So if we're looking at the specific steps, we'll see that the lungs transport the oxygenated blood to the left atrium by the pulmonary veins. And then from the left atrium, the blood is pumped into the left ventricle through the bicuspid valve. And this is like the tricuspid valve, also a valve that prevents backflow just for the left ventricle instead of the right ventricle this time. And then this reoxygenated blood will enter the main artery, the aorta, through the aortic semilunar valve, which again, like the bicuspid and tricuspid, prevents backflow of blood. And then this aorta divides into the previously mentioned innominate artery and aortic arch, which divides into furthermore arteries that supply blood to the brain and the neck and the forelimbs and etc. And then, as stated before, the main aorta will continue as the dorsal aorta to supply blood to the rest of the lower regions that the innominate artery and aortic artery will not reach. Together, the pulmonary circulation and systemic circulation create the efficient double circulatory system that is in the rats. And it's efficient for and advantageous for the rats because it allows for oxygen to reoxygenate blood at a faster rate. And there's also a hepatic portal system in rats, which is when blood is filtered through the liver first through a hepatic portal vein, and this allows materials from the blood to be absorbed in the guts or filtered out to be expended as waste.
Negative human impacts. A major threat to the rat population is habitat loss. It can be caused by energy development from the use of fossil fuels by humans and the pollution of water and air from factories and road spills. Positive human impacts. Firstly, human development, which can include building constructions, farms, towns, and cities, which can allow for rodent infestations and is extremely bad for humans. This is because rats can transmit diseases like hantavirus, leptospirosis, or salmonella, and they can damage human property by chewing through wires, insulation, or concrete. And lastly, they can hinder human food production. Secondly, rat control campaigns, which are initiatives that remove many rats from their populations. However, survivors of these may pass on their fit traits to offspring, overall driving rat evolution.
I now want to talk about our third specimen, which is a squid. The longfin inshore squid, or known by its scientific name, the Loligopili, are invertebrates, and they also are selected species, which means that they prioritize high reproductive rates and rapid population growth in often unpredictable and unstable environments. Now I'd like to move to the squid spine. Different squid species prefer to live in certain living spaces. Some prefer to live in shallow water near coastlines, including bays, estuaries, and kept forests around water, while others prefer to live in the open ocean. The Loligopili, or the longfin inshore squid, prefer to live in continental shelves and slopes, and they reside in the Atlantic coast in the north in Newfoundland to the Caribbean Sea. Adaptations. Squids have many types of adaptations to survive and reproduce successfully, and this includes camouflage and jet propulsion, which are two of the most important adaptations in order for them to survive. These mechanisms include a streamlined body, powerful eyes, camouflage, jet propulsion, ink sacs, and arms and tentacles. The first adaptation of the squid is the streamlined body and the jet propulsion. The streamlined body shape minimizes drag and allows for efficient swimming. Regarding the jet propulsion, it assists in locomotion and circulating water over gills. It allows for effective movement and maneuverability in water, giving them the name the invertebrate athlete. The next adaptation is their powerful eyes. The squid's eye is highly developed and effective and allows them to see in the ocean's diverse lighting conditions properly. The next adaptation is camouflage. Squids can rapidly change their skin color and texture through chromatophores, which are specialized skin cells. This allows them to blend into their surroundings via their various pigments. They have various pigments like red, orange, black, yellow, and brown, and the tiny muscles that surround the chromatophores either contract or relax, which causes the pigment scans to either expand or retract in return, controlling its intensity and color visibility. This mechanism evolved through selective pressures of predators and prey. It also has other roles, such as thermoregulation. The next adaptation is their ink sacs, which release dark ink clouds that are aimed to confuse and distract predators. This in turn allows for the squid to escape, which increases their chance of survival. Their next adaptation is their arms and tentacles, in which they have 8 arms with 2 longer tentacles that are equipped with suckers and hooks, or teeth, to capture prey efficiently. Their next adaptation is reproduction. Males have a modified arm called a hectocolitis, which transfers sperm packages into their eggs. They also engage in courtship displays, which are very competitive and contribute to genetic diversity. The females will prefer to lay eggs in communal egg masses, and sneaker males will try to fertilize the eggs that are laid by others. Their final adaptation is prey capture. Their 8 arms and 2 tentacles contain sharp rings on their suckers to help grasp and hold prey. They also eat prey alive through their beaks, which helps in shredding flesh. Squids will rapidly stretch their arms and tentacles to grasp prey, and this takes about 20 to 30 microseconds. Now I'd like to move on to their ecological niche, more specifically towards their habitat. Squids will inhabit all oceans worldwide, and this spans from shallow areas to depths longer than 16,000 feet. Juvenile squids reside in shallower depths, while adults reside in deeper areas. And some squids, such as jumbo squids, have a specific habitat niche that's influenced by environmental factors. Different squid species are adapted to different types of aquatic environments. Now I'd like to talk about their migration patterns. They undertake seasonal migrations, and they move offshore during the colder months and inshore during the warmer months. This allows them to optimize their environment for growth and reproduction during the year. Now I want to move on to their diet. Their prey consists of small and various types of fish, crustaceans such as shrimp and crab, and other cephalopods, with their predators being larger fishes, seabirds, and whales. Their diet is heavily influenced by their environment, and they also have a high feeding rate and wide range of food sources. Relations with animals. They have a role as predators and prey within marine ecosystems. Their role as consumers allowed them to regulate prey populations while also being an important food source for other larger marine animals. If squid populations are healthy, the predators will also have an ample food source, which in turn contributes to the marine ecosystem's overall health improving. Squids occupy a large range of trophic positions and also exploit a large range of trophic resources. Squids are known to have mutualistic and symbiotic relationships with other animals. Regarding their mutualistic relationship, certain squid species contain a bacteria for camouflage, and certain species of squids, such as the bobtail squids, have a relationship with bioluminescent bacteria. This relationship benefits bacteria and squid as they provide light for camouflage, and squids provide a safe habitat and nutrients. Regarding the symbiotic relationship, female longfin squids have a symbiotic relationship with bacteria in its accessory nidamental gland, or their ANG, which is part of their female reproductive system. And this symbiosis essentially protects the squid's eggs from harmful pathogens. The ANG holds large communities of bacteria which are recruited from the environment. And this bacteria produces antimicrobial compounds which help protect developing squid eggs from pathogens like bacteria and fungi. The squid provides a home and nutrients to the bacteria, while the bacteria protects the eggs, causing this to be a symbiotic relationship. Squids also have a specific role on their ecological niche. They help their ecosystem via nutrient cycling, which is the transport of carbon and nutrients between marine environments. They also have many important roles, as they can be considered as keystone species in the majority of marine ecosystems, and they have a significant role and impact on food webs and energy flow. If squids are not present, that means they have a massive impact on apex predators. They themselves are also predators for a diverse amount of organisms, and the change in squid population means that there will be a change in marine environment. They have a unique and important position in marine food webs, and as stated before, they function both as skilled predators as well as important prey for various animals. And their role as consumers allowed them to regulate prey populations while also remaining as an important food source for predators. Now I'd like to move on to my dissection. The first thing that's shown here on the right is the chromatophores, and the chromatophore is the specialized pigment containing cell that allows for camouflage via rapidly changing skin colors and textures. And it also allows for cutaneous respiration, which is the absorbance of oxygen via water. The next thing that's shown here on the right is the gills or the citonedia, which facilitates oxygen and carbon dioxide exchange between the squid's blood and water, which is also important for respiration. As you can also see, the bronchial and systemic hearts are present. Squids have 3 hearts, 2 bronchial and 1 systemic. The bronchial heart pumps blood for oxygen transport, while the systemic heart pumps this oxygenated blood throughout the rest of the body. In their lower body, they have 8 arms with 2 longer tentacles that are equipped with suckers and hook-like teeth for efficient prey capture. They also have a beak and buccal mass, which is used to break through shells or flesh of prey, and is also used for feeding. Squids have a mantle, which is the main body of the squid that houses its organs. It contains the gills, digestive system, reproductive organs, and much more, and it's also important for locomotion. The siphon is mainly used for jet propulsion, which expels water forcefully, forcing the squid to move. It's also used for respiration and waste excretion. Squids have a large brain and have one of the most complex nervous systems in invertebrates. They have specialized brain lobes in which certain lobes hold the responsibility for memory and learning. Now I'd like to move on to their complexity. They have an elaborate nervous system with a large brain and specialized brain lobes. They have giant axons, in which some of which are the largest in the animal kingdom, and they also have sensory organs with well-developed eyes and sensory receptors. Regarding the respiratory system, the gills, or the citonedia, are a chamber located within the mantle cavity. The gills are bathed with seawater, and then the oxygen will diffuse from the water into the blood, and the mantle cavity, or the water chamber, will hold the gills. The bronchial hearts are responsible for pumping deoxygenated blood from the body to the gills, and the systemic heart will pump this oxygenated blood throughout the body. As stated before, cutaneous respiration is where the skin will absorb the oxygen via simple diffusion. The respiratory system also contains specialized epithelial cells which line the gills and facilitate diffusion, and they're thin, allowing for gas exchange to take place properly. They have a network of blood vessels that surround and spread the gills for oxygenation. Squids contain a closed circulatory system. That means that their blood is confined to their arteries, veins, and capillaries. Squid blood also contains hemocyanin, which is a copper-based pigment that binds oxygen to squid's blood, which also has a blue color. This ensures that oxygen and nutrients reach all the tissues via the systemic heart. How they work together. Mental muscles will contract to create water flow, and the external water flow will lead to gas exchange taking place, in which oxygen diffuses from water into the blood of the gills, allowing for the blood to be oxygenated. And then the bronchial and systemic heart will work together to circulate this oxygenated blood to the rest of the body. Now I'd like to talk about the relation between the organ system structure and their lifestyle. Since they have an active lifestyle, they need a proper respiratory system to meet its needs. The dual function of jet propulsion is important for proper water flow towards the gills as well as locomotion. They use this method for locomotion, ventilating gills, and drawing in water. The importance is that squids are active predators, and that means they need a high oxygen demand, and the continuous water flow via jet propulsion helps with that. Also, smaller gills are more advantageous as water can be rapidly passed in trade for more frequent bursts of speed, and the respiratory system supports the squid's behavior.
Human impact. Overfishing. Squids are especially vulnerable to overfishing as it will have negative consequences for the entire ecosystem, including population size decline, reduced biodiversity, and food web impact. Squids have a large role in the marine food web, and overfishing can disrupt its balance between acting as a predator and a prey for many species. Overfishing can also impact the squid's ability to reproduce and recover. Pollution. Ocean acidification, which is caused by increased amounts of carbon dioxide in the ocean, can delay squid larvae development as well as reduce the size and quality of their satellites. Noise pollution can also disrupt normal communication between squids and ecologically relevant responses to sound. Especially with construction offshore, intense noise can cause alarm responses in squids that can alter their behavior. Metals and chemicals in the environment can also impact reproduction, growth, feeding, and defensive behavior in squids.
An extra dissection that we included for this lab was that we dissected a sheep's heart. And we did a cross-section of this because we wanted to see different features and the anatomy of the heart, different hearts in general. With all the organisms that we dissected, the hearts were pretty small for them, so doing a cross-section between them only gave us so much. But with the sheep's heart, it's significantly larger, so with this we were able to observe different features and anatomy much better. Some things that we noted from our dissection in general was fats and muscle. And the heart has fat because it's its energy source. It basically gives it the energy to pump blood, and it also protects the heart. And a significant muscle that is seen in the heart is a cardiac muscle, and this muscle is responsible for the pumping of the heart. So there's two sides to a heart. When holding a heart in your hand during the dissection, the left and right side can be unclear because you can be holding it in a different way than how it is in the heart. So the left side is always seen through the apex, and this is split by the interventricular sulcus. Another feature of the heart is the auricles, and auricles mean ear flap shape. They're seen on both sides, the right and the left side, and there's a right auricle and a left auricle. And technically it's the atrioventricular groove, which is responsible for separating the ventricle and the atriums. So there's a couple of veins that are pretty important in the heart which allow for the movement of blood. So there's a superior vena cava, and that's a pulmonary vein which from the rest of the body, it brings all the blood from the rest of the body to the right atrium. Another pulmonary vein is the inferior vena cava, and that just brings the blood to the left atrium from the right. So the aorta is another vein which is at the center of the heart, and what it does is that it takes the oxygenated blood from the left atrium and pumps it to the rest of the body. Now between the left ventricle and the aorta, there's also an aortic semilunar valve, and that's what allows for the one-way passage from the aorta to the left ventricle, and it's also three-membranous. So the pulmonary semilunar valve, this is a three-membranous pocket which allows for blood flow from the right ventricle to the pulmonary arteries and the lungs, similar to the aortic semilunar valve as well. All these valves do is that they allow for the passage from blood from one area to another in the heart. So another valve that's part of the heart is a tricuspid valve, and this is flaps of membrane which are connected by papillary muscles. And the papillary muscles are also connected to the atrium and the ventricle by chordae tendineae. So basically what this valve does is it's basically like a gate between the right atrium and the left atrium. So it allows for blood flow between each atrium, but it allows for the blood flow to go one way. So it doesn't allow for backflow so that the blood would leave, oxygenated blood would leave through the heart in the wrong way. So the bicuspid valve is like basically the exact same like the tricuspid valve, except this one's just between the left atrium and the left ventricle, while the tricuspid valve is between both atriums. Specialized cells that are in sheep's hearts are myocyte, endothelial, and fibroblast. So myocytes are specialized muscle cells. These are involved with the heartbeat contractions, and they're generally the most abundant so that they can help out in general. They're like the cardiac muscle, but it's more specific. And there's also the endothelial cell. This is the inner surface of the blood vessel, and this contributes to vascular health. So it's involved with like regulating the blood flow, keeping the health of the vascular health up. Finally, there's the fibroblasts. These are involved with tissue repair, and they're the structural support by producing the extracellular matrix. The disease that we chose to research is chronic pulmonary obstructive disease, commonly referred to as COPD, and is primarily a respiratory disease but can also affect your circulatory system. So common symptoms of this, generally when you primarily get it and it's not as severe, will be a daily cough to get your mucus out, wheezing, chest tightness, and as your symptoms get worse, there's lack of energy, swelling in feet and ankles, and then commonly weight loss. Generally, why COPD occurs is because of inflammation or swelling of your air pathways. So this can be your trachea, your bronchi. And primary causes to this damage are usually being in harmful environments. So this can be if you are exposed to fumes, smoke, or if you smoke yourself, like smoking tobacco. So there's a couple of common types of COPD which can occur. There's emphysema and chronic bronchitis. So emphysema occurs when your alveoli are damaged, preventing oxygen flow to the rest of the body. So the alveoli they're small sacs in your lungs, and if they can't properly store good oxygen, then good oxygen can't go to the rest of your bloodstream. Chronic bronchitis is the inflammation of your bronchi. So that's one of your airways which take in air, and this prevents good airflow from coming into the lungs, which also creates mucus in your throat or in any of your airways. Both can occur together, and the severity of both can be determined based off the person. So one isn't worse or better than the other to have, and they can both cause a lot of damage or not as much. So general complications that come along with having COPD or can happen after you even recovered from it are higher chances of getting respiratory infections. So this can be like colds, flu, pneumonia, and also your chances for lung cancer become a lot higher. Also, it can affect your circulatory system because you can have higher chances of getting heart disease, plus in your lung arteries, there's high blood pressure over there as you get COPD more, or you can have high blood pressure after getting it. And this is called pulmonary hypertension. So possible treatments that you can get for COPD is when it's in the primary part of having it and it's not as severe, you just have to improve your quality of life. If you smoke, you have to quit smoking. And if you're in areas which have high fumes, smoke, generally you have to stay away from them. But as it gets worse and more severe, generally you have to take certain medications, or you can do pulmonary rehabilitation or oxygen therapy.
We also decided it would be beneficial to compare the evolution of the respiratory and circulatory systems of the organisms that we decided to dissect, as well as how they adapted to their environment as the environment changed over the years. The first organism we dissected was the frog. Originally, frogs were actually fully aquatic amphibians, meaning they only lived in the water. Their primary source of respiration were their gills. However, now that their environments changed to swamplands, ponds, and forests, they had to develop a new way to breathe due to being on land more often. So, due to natural selection, frogs that could get oxygen from both the water and the land survived, while the frogs that could only get oxygen from the water died out. They ended up developing small, simple, sac-like lungs to help them breathe, which is important to the frogs in their adult stages because when they turn into adults, their gills and their tails disintegrate. so they have to develop lungs. They don't have the complexity of normal mammal lungs, and so respiration through their skin is also really important for them. This is known as cutaneous respiration. The larger surface area to volume ratio for the skin is really necessary for the frog because it helps with oxygen transfer, and the skin is also a thin membranous tissue that has a lot of blood vessels in it that helps with like blood flow. The skin also has mucus glands that can help the frog maintain its moisture outside of the water and prevent it from drying up, as well as absorbing additional dissolved oxygen. Now moving on, we dissected the pigeon. Originally they were found on coastal cliffs, rocky terrains, and near cliff edges. However, now that humans moved into their habitats, they can now be found in more urban areas like cities and agricultural areas. Pigeons actually have a unidirectional airflow, meaning they take in new air every single breath they take, as opposed to humans that have a bidirectional airflow, and so every single breath they take mixes with old oxygen that's already stored in the lungs. The unidirectional airflow system of the pigeon helps them gain more oxygen from their surroundings, which is really important for them because they need the oxygen to generate energy for metabolic processes as well as during flight. They also have 9 air sacs within themselves that help them store oxygen for later metabolic use. Due to domestication, however, they lost their original habitat and so they had to adapt by many ways. One of the ways they adopted was their housing ability. So they can house in like various different locations, like on windowsills or on building ledges. And they can also scavenge for food in the streets of like the urban cities. And they can also peck at like the sidewalks and stuff to find seeds, etc. They also developed behaviorally as well. So as opposed to the past, now pigeons are a lot less scared of humans because they're exposed to them so often. They also changed physically and with their features as well. Depending on their habitat, pigeons can take on different beak sizes, feather patterns, and sizes, which helps with sexual selection. As you can see on the image on the bottom, there are many different colorations, patterns, and sizes of pigeons across different habitats. Next, we dissected the rat. So what's interesting about rats is they can be found in many different environments, including originally they were found in forests, fields, islands, riverbanks, woodlands, and tunnels. However, now that humans have taken over much of their habitat, they can also be found in garbage dumps and sewers and subways. So rats come from heavily human-populated areas, and because of this, they can show significant signs of evolution. In the human-populated areas, they're exposed to more pollution, and so they have increased exposure to harmful gases such as CO and SO2. The rats had to evolve to have many structures within themselves to filter out harmful particles within the air. So one of the things that they evolved with was a secretory cell called hyperplasia, which basically thickens the mucus and traps harmful particles within that mucus. Furthermore, they had to adapt by being able to nest in unconventional areas that humans would usually reside in. They also developed a specialized Stennios gland in their nasal cavity, as well as an increased abundance in ciliated cells. And basically what both of these features does is it filters out larger and more harmful particles and protects the airways of the respiration system. If we look at older rats, like older rat species, they don't have as many ciliated cells as they do currently. Some of the things that they adapted to behaviorally wise was an increased neophobia, which is basically a fear response to new stimuli and an avoidance of new objects. This is incredibly important for the rat because now that humans have occupied their areas, humans see the rats more as pests, and so it's just an added danger to the rat. So it's always better for the rat to be more cautious of their surroundings. So some morphological adaptations were they they now have longer tails that support thermoregulation, as well as a thicker fur for them to keep warm in colder climates. Now, they also have an internal jugular vein, which is not that useful to the rats in their adult lives, but however, in the embryonic stage, the jugular vein helped increase blood flow in the embryo. They also unfortunately lack well-developed respiratory bronchiolitis. Next, we have the shark. So, originally sharks were found on the top of the ocean's surface. However, as of now, they can be found on the bottom of the ocean seafloor, as they are beniopalactic. This basically means that they reside on the bottom of the ocean seafloor as well as they feed on the ocean seafloor as well. So, because they're on the ocean seafloor so often, they don't really move around that much, and so it's harder for water to circulate around them and get through the gills. This is what the spiracle is used for. So basically it's a vestigial structure that can be seen on the side of the eye of the shark, as you can see in the picture presented. So basically the spiracle helps draw water directly over the gills and it helps the shark further be able to take in oxygen from the ocean. Something interesting is, in primitive jawless fish, the spiracle was the first gill. As jaws further evolved, however, the spiracle became separated from other gill openings and it developed into its own specialized structure, which proves evolutionary transitions from jawless to jaw-bearing vertebrates. This is so important for the shark because it allows them to regulate the gas exchange while minimizing water loss, which can be often seen in organisms that rely on their gills for respiration. This also conserves energy so the shark can hunt and eat simultaneously. Something interesting that we found was embryonic and fossil evidence can prove that the human middle ear actually evolved from the spiracle of the spiny dogfish shark. The next animal we dissected was a squid. So originally the next organism we dissected was a squid. Originally, squids resided in shallow, low-latitude shelf areas of the ocean. However, now they can be seen in more open ocean environments. So, squids actually evolved from mollusks, as you can see on the pictures on the slide. They utilize their gills for respiration just like how a mollusk would. They have closed circulatory systems, multiple hearts that reside in their mantle cavity, and copper-based blood. Their respiration system as a whole is really important for them because it helps them in different oceanic environments, and it also helps them adapt to these environments that may not have as much oxygen or it may have faster moving water. So early cephalopods actually had simple gills, and over time squid gills evolved into having many folds within themselves. The folds actually increase the surface area to volume ratio of the gills and helps for oxygen to diffuse within the squid a lot easier. So they also evolved their closed circulatory systems, which allows for a more rapid and efficient blood flow within the squid itself. Now moving on to the earthworm. Something pretty interesting about them is they have thought to originated from completely aquatic environments, but now they can be found in more terrestrial areas such as soil and leaf litter. So because they evolved from aquatic organisms, early earthworms can be seen to have respiratory structures similar to gills on fishes. However, now they don't have any respiratory structures because they can be found in soil and more terrestrial areas. So they completely rely on cutaneous respiration for oxygen diffusion, which is basically oxygen diffusion through their skin. Early marine annelids likely had circular systems that relied on general diffusion, whereas now earthworms have a more closed circulatory system with vessels and pseudohearts. Something pretty interesting about the earthworm is that because they have multiple hearts and their main organs are in the top half of them, if you were to cut them in half, they would still be able to survive and regenerate. Basically, this closed circulatory system is the most ideal for underground movement, because it enables a better blood flow, which is more efficient and direct. Lastly, our group decided to dissect the sheep heart as an additional dissection. So, originally sheep were found in mountain ranges, plateaus, and arid environments. However, as time moved on, they can now be found in other environments such as uplands, plateaus, grasslands, and deserts. Sheep were actually raised for domestication, and so because humans raised them to shear their wool and sell it, they actually lost their ability to naturally shed their fur. If you were to see a sheep in the wild, their wool would actually keep growing, and so this could actually be harmful for the sheep because it can prevent them from doing their daily activities, and it can actually be harmful because the sheep can overheat because of this excess wool. They have a 4-chambered heart, which evolved from the 2-chambered heart. This newly evolved heart can allow for more efficient separation of oxygenated and deoxygenated blood. Furthermore, they have a 2-lobed left lung and a 4-lobed right lung. This right lung is actually more efficient than most mammal lungs because of the greater surface area to volume ratio that the 4 lobes gives the lung. The greater surface area to volume ratio can further help oxygen and CO2 exchange. They also have a single large common jugular vein, which is located in their neck. It's the main drainage route from the head and the neck. So this internal jugular vein is either extremely small or completely absent in certain sheeps. Lastly, they have the carotid rete, which is actually a developed network of small arteries in the neck which can help regulate brain temperature. This is so important for the sheeps because, as mentioned earlier, they can be found in drier environments such as deserts. And so this carotid rete can actually help keep their brain cooler in these humid and dry environments.
Now we'll show our cladogram, which depicts the evolutionary relationships between all of our organisms. All our organisms branch from one common ancestor. The shared trait between all of them is that they all have a closed circulatory system, which means that blood in every organism travels through a network of closed blood vessels. Our first divergence in the cladogram is between vertebrate and invertebrate organisms. This is the point where we start to see the main differences in their respiratory and circulatory systems. Our 2 invertebrates, the squid and the earthworm, share an ancestor called Lophotrichozoa, which is an ancient organism that appeared 542 million years ago and is characterized by its tentacle-like body. The earthworm falls into the phylum Annelids, while the squid falls into the phylum Mollusks. We also have 5 organisms that are vertebrates: the sharks, frogs, pigeons, sheep, and rats. Ascending the cladogram, you'll find each distinguishing feature that separates all the animals above from the animals prior. Frogs and so forth are tetrapods, meaning they have legs and are land creatures compared to our shark, which has fins and lives in water. Pigeons and so forth are amniotes, which means their embryos fertilize in membranes, which for pigeons are amniotic eggs and for the rest are amniotic sacs. Lastly, we have our sheep and rats, who are mammals and unlike the others, which means they are warm-blooded and nourish their offspring with milk that they produce. Now we'll focus solely on our circulatory and respiratory system. As the organisms get more complex, they begin to have larger numbers of heart chambers and lung lobes. For example, our earthworm has merely 5 aortic arches, which aren't even exactly full hearts, they're pseudohearts, compared to our pigeon, which has 4 whole chambers and more lobes for its lungs. Furthermore, single circulation, which is when blood only passes through the heart, seems to be a feature that arises from convergent evolution, as the sharks and squids aren't evolutionarily related other than their common ancestor. However, they've both evolved single circulation as well as gills due to their aquatic environment and lack of lungs. Earthworms are similar to them with their lack of lungs, as they instead breathe through their skin. Another feature that may or may not be from convergent evolution is the fact that earthworms have double circulation, which is commonly only seen in vertebrate organisms. Double circulation is when blood passes through both the heart and the lungs, which for the earthworm it instead passes through their digestive tract since they don't have lungs. When we look at the more complex organisms, we see a difference in their heart chambers and lung lobes, which signify the efficiency needed both due to their bodily structures and functions. Frogs are smaller, which explains less of a demand for oxygenated blood from its hearts and lungs, and why it only has 3 chambers and 2 single-lobe lungs. Pigeons and the rest have 4-chamber hearts because they're the most complex organisms. As pigeons have to fly, sheep are far larger, and rats are very speedy. The difference in lung lobes can also be explained by their functions, as pigeons would need more efficient air circulation when flying. Sheep, again, are larger, which would mean they need more oxygen to function in general, and rats have high heart rates and thus pump more blood, which is needed to sustain their fast lifestyle. Overall, the evolutionary relationships between this organ system and these animal lifestyles are a result of the evolutionary history amongst the animals themselves.